Hi, my name is Emily Baugh, and for a group assignment project, we decided to do genetic editing. To start out with genetic editing, it is a way of making specific changes to DNA of a cell or organism. And it is when an enzyme cuts the DNA at a specific sequence. And when this is repaired by the cell, a change or edit is made to the sequence. In other words, it is used to alter human genomes. And it is of interest because genetic editing can be used to cure diseases like cystic fibrosis, which of now there's no cure or specific treatments for it. And it could also be used to delay or stop aging. Also, genetic editing can be used to help researchers find treatments for the diseases and help understand the diseases more. And there's also talk about how newly parents can alter the genes that their unborn baby could possibly have by picking out which characteristics they would like their child to show. For example, like blue eyes instead of brown eyes, they can pick which favorable genes that they want their child to have. And that is it for genetic editing. So now I will direct your attention to Monica for information about how the current state
My name is Monica Gonzalez, and I will be discussing a current state of knowledge in genetic editing that I found quite intriguing. Genetic editing may appear to be safe at the moment, but a new study early this year warns us about the long-term effects of these practices. This new study clearly illustrates the cancer-causing potential of genetic editing. Experts have said that CRISPR Cas9, which entails altering specific gene in cells, could help overcome diseases such as cystic fibrosis, HIV, and Alzheimer's. Now, this almost seemed too good to be true. Nature Medicine has printed 2 studies that show how the treatment could actually trigger cancer. Emma H., one of the authors for the study, has explained how the cells with genomes that are edited successfully by the procedure can actually seed tumors inside of people. This problem seems to apply when CRISPR is used to replace DNA that causes diseases. This is something that needs some serious attention because, after all, the goal of the procedure in the first place was to avoid disease. It doesn't make much sense to return edited cells to people that are likely to become cancers. And now let's move on to any controversies with Janine.
Hi, my name is Janina Spino, and I will be presenting over the controversy surrounding genetic editing. The high level of interest in genetic editing comes from the potential it has to transform a variety of scientific areas, from treating medical conditions such as Duchenne muscular dystrophy to eradicating some genetically inherited diseases such as Huntington's disease to agricultural applications such as improving crop yields and resistance to pests. Genetic editing is and remains an all-around controversial issue. While it could potentially spare future generations in a family from having a particular genetic disorder, it might affect the development of a fetus in unexpected ways or have long-term side effects that are not yet known. There have been concerns about off-target effects, where altering the DNA sequence in one part of the genome may have unforeseen consequences on another part of the genome. In summer 2018, 3 papers were published which raised concerns about off-target effects. 2 of these papers reported that cells that had successfully been edited using CRISPR-Cas9 turned out to have a defective p53 gene, suggesting that CRISPR cells might have the potential to cause cancer. Because of these ethical concerns, the U.S. government does not allow federal funds to be used for research on germline gene therapy in people. However, the most controversial issue surrounds the potential use of CRISPR to create genetically engineered humans. In 2015, following the first international summit on gene editing, the consensus was that there should be a global prohibition on heritable genome editing in humans until further research had been carried out into the potential risks. The news that rocked the scientific community was a Chinese researcher, Hai Jiankui, who had apparently successfully used CRISPR to create gene-edited babies, twins actually, resistant to HIV infection. His research has yet to be published or peer-reviewed, and there is limited information about how successful the technique has been. His presentation and the discussion following only raised more questions than it answered.
Hi, my name is Haley Simpson, and I'm covering the topic of the future possibilities for gene editing or genetic editing. So according to healthline.com, there are research and things going on right now to figure out ways to have what people are calling designer babies, where, say, the parents want them to have more athletic abilities or be more intelligent or have a certain looks, and these are the ones that are more controversial. But there are other possibilities for genetic editing, such as curing genetic diseases or creating a new population of people who are resistant to these diseases. So those are the kind of studies that are going on right now for genetic editing. But there are also hopes to be able to edit the genes of adults and children who are already alive, and taking away that ethical concern that so many people have. That's all I really have to say on this topic, but the possibilities seem endless with genetic editing. So let's see what the future holds.